how do we move from whatever understanding we have of what the strengths and weaknesses are of these methods to actual decisions that matter? So it seems pretty clear that we're going to spend some money connecting people's records and being able to have the infrastructure for observational studies. It seems pretty clear that we're going to fund some statisticians and their ilk to develop methods that help us better understand these limits. Those, to me, aren't really questions. It seems obvious we're headed there. Maybe there are questions about how much. It seems to me the real question PCORI faces is, will it ever spend large amounts of money on really big RCTs? And I suspect I know the answer to that, too, which is I'd be a little surprised right now because I haven't seen a hint that they're going to do it. Now, maybe that's not true, but even if it is true, it raises another set of questions, which is how do we actually make prospective decisions about really big investments to decide when to make them? What tools do we use? Do we look at things like value of information analysis? Do we convene expert panels? So I guess I really want to have us spend some time thinking about this question. Because at the moment, the most obvious question to me is, will PCORI fund or will other entities fund really large trials and under what conditions? So that's my...
Okay, great, thanks. I hope somebody's writing these down, by the way. I don't have a pen up here. Sally, did you want to, okay, Bobby, and why don't you do that and identify yourself and where you're from and then what you
Bobby
hope.
Du Bois, the National Pharmaceutical Council. It seems to me there's the two ways, it's sort of the old lady, young lady picture. And one aspect is, how do we design a study to answer a question? So most of us in here are scientists and researchers, but the young lady one, or old lady one, is if I'm a patient and a doctor and a study has been done, do I accept that as something that can guide my therapy? And there are different criteria. One is sort of optimally, how might we design it? And the other is the sort of reality that here is a study, is it worth consideration? And the choices you make might be different on both sides of that divide. Something I suggest maybe we can keep in mind.
Thank you. Okay, Sally seems... Okay.
Hi, Mark Berger, Pfizer. One contextual comment. Regardless of what we think is going to happen, the volume of observational data is going to dwarf by orders of magnitude the RCT data coming out. So everybody talks about big data, and it's here clearly in commercial and in supply chain, but the era of big data is upon us in healthcare. So it's not a question about whether we're going to use observational data, it's about how we're going to use observational data. It seems to me, though, that the real issue is not whether we can come up with the best way to decide what the right design is and understand the strengths and limits of it, because as researchers, we should be able to do that. But decision makers oftentimes don't know what is exactly the question that a study is answering, because even in the publications, the researchers themselves don't delimit the exact question that they're asking. So they don't know how to put a study into context. And that's a real problem. A third issue is that regardless of how good the data is and how good the results are, decision makers are making decisions, and they're making decisions off of the research that's being generated. They don't have the luxury to wait saying, well, this is not so good and that's really good. They look at it and in some manner or form they synthesize it together and they make decisions every day. So it's up to us to make it clearer to them about what are the questions that are being asked so they can put that data and that information into better context.
Thank you.
So I'm Sally Morton, University of Pittsburgh. I think we have to remember that we're not thinking about single studies here, but rather looking across a group of studies. And in fact, a lot of the data that are being generated that our speaker just alluded to aren't generated from a study. they're generated without a design study being put in place. And that's a very different place to be. We think about this in statistics, all the Google data and, network data. And so it's sort of backwards in a sense. There isn't a design and then data generation, but data just appearing. And how do we work with this? So I think we really have to think about synthesis across studies. It's my first point. The second point is that I think it's a great time right now. I think we shouldn't lose this moment. I really feel strongly about this, and I think that PCORI is a wonderful vehicle to really get out and communicate, educate, and engage methodologists as well as stakeholders. And so we have to use this moment wisely. My hope for this workshop is that we do come out with practical steps, as you alluded to in your last couple slides, Steve, of what we should do next, both in the short term and the long term.
Thank you.
I'm Joel Greenhouse from Carnegie Mellon University. I guess I'm following up on Sally's comments from the Pittsburgh connection. Steve, you asked why scientists disagree about the strength of evidence. And this is certainly an interesting question, but I'd like to turn that question a little bit and ask the question, shouldn't we be thinking about what is required to bring the scientific community to consensus about an important question? It's got a slightly different approach, but I think we've neglected that. And I think I'd like to see the conversation move towards that, because what is the sort of evidence we need? How can we move in that direction so that we do have consensus? Not everyone's going to agree, but can we reach a majority consensus? And I'm also reminded, I'm also a big fan of Sir Bradford Hill, that in talking about clinical trials, he sort of considered a situation where a mortality rate was 80, 90%. And he said, in that context, we don't need to have a control group. We'll know the answer if we see a treatment effect. And so we need more wisdom like that instead of just sort of falling back on sort of set methods. And so again, I want to come back to the question, and I hope we can maybe address that. How can we reach consensus on important questions?
Thank you. That's a good point.
Steve, just before the next question, we just got a note. People are listening on the web because we got a request for speakers here to speak more directly into the microphones. They're having trouble hearing somebody.
Thanks for the heads
Okay.
up. Anirban Basu, University of Washington. I liked your point about uncertainty that you brought up, and I was wondering, there'll be a discussion about what's the acceptable level of uncertainty for individual level decision making. I mean, we always think about type 1 errors, but is there an acceptable level of type 1 error? Because here we are trying to inform decisions and mostly decisions at the individual level. And so how do we set those kind of uncertainty, acceptable uncertainty from our research?
Thank you. Jamie.
I'm Jamie Robbins from Harvard School of Public Health. So I had an interesting day yesterday. I was at the Microsoft Machine Learning Summit in Paris. And
What time is it for you right now?
even worse, I left my computer and cell phone in the airport, and you can't get anyone on the phone. But that's another question. Or the internet. As you all probably know, over years I've worked on these causal methods, and there are a lot of strange methods out there that some people use and some don't. And what we've always tried to do is, when we use these methods, like analyzing the effect postmenopausal estrogens, compare what we get with randomized trials, and we do more complicated things, like trying to figure out the effect of when you should start therapy for HIV-infected patients. We compare with the few trials there are, but there are few examples because there aren't many trials. Well, at Microsoft, they've started using these methods, exactly these methods, because they have a huge time-dependent confounding problem. Their big problem is, given the person who's on the computer at the moment, which unfortunately they know about everything about you when you're on the computer, how much do you spend knowing you to put what ad at how high a level on those things that come up. And so it's exactly the doctor's problem. Someone walks into your office, this patient, you know all these covariates about them, including how they've reacted to treatment, what you had in the past, and your problem is to make this decision. And so the whole issue is they aren't so worried about the null hypothesis, which often we are. We don't even know which side it's on. Their question is, what's the optimal treatment strategy? And they've even run a bunch of randomized trials, but just like us, the trials are far from the complicated regimes they're actually interested in. And the trials have been a week or two, and things change, and they have trillions of observations. And the bottom line is they've started using all these doubly robust, inverse probability weighted marginals, these kind of things. And everybody was at this guy's talk. This was, heartwarming to me. They're making billions that other people are not making. And it is the hottest thing there is. And the point is, and if you think about the way they think about it, they continually are getting new data. They continually are changing the optimal treatment they're doing today based on results they're getting in, plus they're constantly doing exploration. They constantly put enough randomness. Not everybody they try a lot of different strategies to see which ones are working. And as you move further away from the ones people are currently using, if people have used these things, you have very few people have followed the decisions you'd like to try. Those are equal to the big inverse probability weights you get when you do like marginal structural models. And so all they do is they design things to move closer, to have more people do things close to what they want. Now this is an impossible dream for medicine, but it's probably good to have a role model out there of people who get as many patients as they want, can do as many randomized trials as they want, and really care about the individual specific treatment regime, because it's really the bottom line. And so it's the idea, if you had in the future all these databases together, had them all merged with randomized trials, could use these correct methods, and were allowed not to make this decision this day that we should ban this or use this and this, but actually have a community-wide exploration of, we're not sure about this in real time as they're doing it. Why don't we try something, why don't we try analyzing it this way and ask the question hypothetically, this is what these methods do. If you use this treatment strategy, what would we have seen? We're not using it now, so it's a counterfactual. And then if it looks good, you actually try it a bit. And this will never happen, but what's interesting is it is happening at this moment, but not with human patients, but with your clicks on the internet.
Interesting. So I should have earlier, when I mentioned Miguel's paper, of course, Jamie was a very important part of that team. I didn't see you in the audience. And I will also add to what you just said, that I have seen over the past year, particularly being at Stanford, a tremendous, a large number of presentations about coming from the informatics world, about the way they look at data, and I keep thinking epidemiology just doesn't know what's coming. Do you want to comment? Sure.
Jamie, can I just ask you to follow up for a moment? Because can you apply this way of thinking to the physician who has a patient in front of her for whom she needs to make a clinical decision? So she is looking for a match, an approximate match of the patient that she sees, but in a very large universe of information about similar patients and how their treatment was affected the clinical course of disease. So how would we think about applying techniques like that to clinical decision-making at the bedside or in the office?
Well, if you think of it in the strategy I was talking, there are certain things you can say now, given a patient's covariates, from trials and this kind of research, that you can say at the present. So for example, if you want to start therapy with HIV, when to start therapy on an HIV-infected patient, we have enough data and enough good analyses to say somewhat about the CD4 count you should start at. But you say, well, but my patient has had this HIV RNA thing, they also have hypertension, they have this and that. We don't have enough fine-tuned data. I mean, we could put models, but no one would, I mean, these marginal structural models, you can model these things and hope you know it's going to extrapolate linearly from what you have. But that's the whole point. At any moment in time, you've only got enough information to effectively utilize some covariates. And instead of saying, oh, we should be able, which I often hear, oh, we should give this patient totally personalized medicine, we should realize at a given point in time we know enough to give some personalization. Your CD4 count. We can't personalize things we don't have enough data or haven't done the analysis on. And so we can tell a physician something. He says, well, I have all these extra problems. Say, well, we've done this analysis, maybe, but we don't believe it yet. Let's try to get more data.
Thanks. So we have about 7 more minutes. Hopefully that will be enough time for Bob and Hal. And
I'll try not to use them all. It would be very helpful to have examples of the inability to generalize to a real population from the results of clinical trials. I have trouble thinking of too many examples of where that's true. And it's interesting that the concept of the large simple trial put forth by Peto reflected the idea that in general the direction of effects in all populations are the same. So they didn't want to look at specified people, they didn't want to rule people out, they didn't want to get a whole lot of information about who they were because they thought it was going to be directionally the same all the time. There's been a lot of movement away from that because we know about genomic factors and stuff like that, so we're all excited about it. But it would be very helpful to know what it is we think we're missing from the large trial. I hear the assertion over and over again that it's not a real population, but where has that turned out to be important? It would help to know.
One thing I'll just say is I think in the realm of drugs, and to some extent devices, it's very different than the full range of interventions that Pecori is interested in, many of which are very context specific. And I think the more context specific the medical intervention is, or the more levels of systems of care it takes for the effect to be optimized, that's where you get the problem. So I think drugs arguably are the area in which you have the least context dependency. It's certainly, you need drug monitoring, you need dosing, all that. But as you get away from drugs, I think you see this more and more. So I think I accept the comment and to some extent agree with it in the drug realm. But I think we find when we get away from drugs, it becomes increasingly complicated. But hopefully we'll explore that.
Well, you know, it would be, I mean, if I were telling Pecori what to do, I would say use a wide variety of ways and as intelligently as possible to form hypotheses, and then at least in a few cases test them, see if they're true. I mean, everybody likes to quote people when they say what they want. Jerry Kornfield also said that a hazard ratio of less than 3 is incredible in an observational study. Well, maybe 2, maybe 3, whatever.
Is not credible.
Is not credible.
Okay,
so
not incredible.
That was a very high standard. And the things we're talking about, you're not going to have hazard ratios like that at all. So it's very much worth thinking about. The other thing I'd put forth, I wonder what you think about it, is if I were making observational studies of something, I would always try to replicate it in a different setting. You know, do one in this group, then do the other one in Medicaid. See if at least you can show the same thing, preferably before you know the answer on the first one. Anyway, the last thing I wanted to say is about individualization. We all believe in it. The genomic revolution is really provoking us to think that there are lots of cases where that's true. But where you don't have an explanation like that, it's hard to know how to individualize. One of the things that one of my favorite studies is to take people who fail to respond to one drug and see if you can get them to respond to another drug. I only know of 4 trials ever that have been done that way. one of the most interesting, reflected the view of all rheumatologists. They know people respond differently to NSAIDs. They know it. It's obvious, and patients come up and tell you that too. So Merck did a really good study, not published as far as I know, but they did a really good study. They took Celebrex non-responders with osteoarthritis and randomized them to Vioxx and Celebrex, which, you know, who knows? Vioxx could still be with us if that had won. There was not a hair, not a paper thickness difference between those two groups. Both groups responded exactly the same when every rheumatologist in the world would have told them differently. And I would have too. I would have believed it, but it wasn't true. So we need to find these things out.
Thank you. Al.
Al Sox from Dartmouth. I wanted to underscore what you said, Steve, about trying to relate the strengths and weaknesses of the research that generates data for patients to make decisions to the actual making of those decisions, which in its ideal form, I think, means having the benefits and the harms laid out and then trying to make a decision. So I'm hoping that one of the things we can pay attention to is how do we lay out the information that we generate with all its uncertainties so that patients and doctors can achieve their decision about, for them, what is the right balance between harms and benefits.
Thank you. Yeah, one point I think I maybe didn't make that clearly is often the designs that generate the best evidence about benefits versus harms are quite different. So it isn't always the same study that gives you both.
Yeah, the next session like this that you're going to sponsor, Joe, is going to be about how you decide about the benefits and harms and what the balance is.
Yeah, great. Thank you.
Good morning. Question from the web audience. Rick Curro at the Pearl Network, New York University asks, what are the common elements between observational studies and RCTs that can be used as a basis for regulatory submission to ensure data integrity?
Well, why don't we write that down and perhaps see if the sessions coming up will address that. I think it's too complex in the last minute of this session, but let's not forget that and make sure that Bob responds to it. Mitch, you get the last word.
Mitch Gale from NCI. Just a couple points. I think it was Dr. Meltzer wanted to know about the potential of clinical trials as part of this portfolio, and it seems to me that for some of the questions that we're trying to address, such as usefulness in subgroups, it's the clinical trial and maybe the follow-up and analysis of the clinical trial that has the most helpful information. So I'm hoping that we'll think of the clinical trial not only as a means to address a particular simple question about a main endpoint, but also to learn about, as has already been stated, the variety of endpoints, the effects on the variety of endpoints, and also the interactions between treatment and subgroup that might give some information on usefulness for subgroups or particular types of patients. The other, but thinking about observational studies and what they can do in conjunction with the clinical trial, I think Hal brought up the point of risks and benefits. And the clinical trial might tell you what an intervention does for various outcomes. But if you really want to understand the way those risks and benefits, you have to know something about the baseline levels of risk for people with various characteristics. And I think the observational studies can be used in that context together with clinical trial data, to get a much more important understanding of the weighing of risks and benefits. And I guess, well, there are other things that I hope we'll address as to the particular needs of observational data if you're going to try to use it, which I think is very difficult to learn about the effects of interventions. But if you are going to try to do it, what are the particular features of observational data that you have to concentrate on? Miguel's paper was alluded to several times, but one feature that he took advantage of was that there was longitudinal information on precisely when certain people began to initiate a treatment. Do our observational databases allow that degree of discrimination to make analyses of this type possible? Thanks.
Thanks so much, and thanks everybody for fitting within the time. I would say that one of the things that the planning committee thought was extraordinarily important in this meeting is to get folks together who spend their life doing observational research and clinical trials with many of the methodologists and statisticians who developed the methods that they're using. There's not enough crosstalk between them, and I think we're seeing just in these comments here the value of a conversation that hopefully will happen over the next day and a half between these two communities. Anyway, that's it for this session. Thanks so much for that, and we'll roll right into the next session, right? Cool.
We are going to move right along, but not without thanking Steve first for a marvelous kickoff. That was a great preview of the day and a half to come. So I'd like to introduce next Dr. Michael Lauer. Michael is the director of the Division of Cardiovascular Sciences at the NHLBI. He is also a member of the Methodology Committee, and Michael is going to moderate this first session on the issue of bias and reducing bias in observational studies.